Restriction digestion is usually used to prepare a DNA fragment for subsequent molecular cloning, as the procedure allows fragments of DNA to be pieced together like building blocks via ligation. Restriction digestion is accomplished by incubation of the target DNA molecule with restriction enzyme. Enzymes that recognize and bind specific DNA sequences and cleave at specific nucleotides, either within the recognition sequence or outside of the recognition sequence. Restriction digestion can result in the production of blunt ends, the ends of a DNA molecule that end with a base pair, or sticky ends of a DNA molecule that end with the nucleotide overhang. The restriction enzyme digestion takes advantage of naturally occurring enzymes that cleave DNA at specific sequences. Enzymes like EcoRI cut through the DNA strands at nucleotides that are not exactly opposite each other. In this simulation, restriction enzyme was used to digest plasmids in specific location to isolate areas of interest. Change the size of plasmid and where the enzyme will cut the DNA, the length of the fragment that will be produced can be predicted. The aim of the experiment is to produce two DNA fragments. One fragment contains a gene of interest and the other fragment is the plasmid backbone that can be used to express protein in bacterial cells. The same two restriction enzymes used for both plasmids. It ensures that the fragments have complementary sticky end that can be joined together. The buffer solution is a solution tube containing buffer at 2.5 times concentration for the restriction enzyme digestion with purple labeled. It functions as providing the optimal pH environment for enzyme function. The restriction enzymes is a solution tube of restriction enzyme PEMHI and HIND with black label. The pKANR plasmid carries RFB gene promoter to control gene expression, TBAD, and a gene that makes bacteria resistant to the antibiotic kanamycin CANR. The PARA plasmid contains a gene that makes bacteria resistant to the antibiotic ampicillin MR, and an arabinose activator RAC that activates the TBAD promoter when the bacteria are grown in the presence of arabinose. The distilled water added to control K- and A- tubes, but not the experimental K+ and A+ tubes, because to keep the volumes of the experimental and control tubes consistent. In order to confirm the result of the restriction digest, the gel electrophoresis was used to separate and verify the size of fragments generated by the restriction enzymes.